Guys, that's not cool. These cost 30,000 each.
Was there a reason why they chose the automatic versus just doing traditional?
Kids were messing it up.
No.
And what is this?
This is what this is where the waste. It's going to run through each one of these stains in sequence. It's all programmed and timed, and that's the waste container because we can't. There's been some complaints about the stains going into the sink and whether or not it harms the microorganisms in their waste treatment plant. So.
Justifiable, I guess.
There was an incident where someone dumped something down a drain. All the lagoons over there turned a color, and all the microorganisms died. They didn't know if it was chemistry, art, or us, but they basically said we don't want anything dumped down those drains. So we collect them. We actually have a company that'll come collect these. All right, so that was the crystal violet, and now it's going to run over to a rinse cycle. So now it's filling up the water base. I should have had everyone come back. We should have just done that.
We're not putting it back onto the slide now. We're just looking at it from.
So now it's going to rinse. There goes all the crystal violet.
Oh, how cool.
Oh, wow.
And it'll sit there and again bob. So this is what you would have done manually. You'd be tipping your slide up and running the water over it. And now it'll do it all for us.
Huh?
Yeah.
They should make this for humans. Make sure everybody's clean.
All right, run through. Yeah, everyone line
Everybody
up.
has to go through.
Lay down crystal violet.
Just dip.
Well, that's why a lot of those places do say take a quick shower before you get in the water. The idea
It's
is
just like you go to the gym. You're supposed to take a shower before you get in the water, before you go in the jacuzzi, before you go in the
Okay.
Now I'm a little shocked at how dark those are, so we will see if this works.
So it's not sitting in that big like petri dish.
Okay, so it's going to drain. So now it's going to do the iodine. This is called the mordant step. Iodine complexes with crystal violet forming a large crystalline structure that locks the stain in. That's pretty much what this step does.
We don't have to know how to do this, right?
You do. Well, you just have to do the steps.
press the
You
play
have to
button.
know it. You have to know the sequence of
I'll put it on
the steps
YouTube.
and the purpose of each step is what you have to know. Yeah, I should call everyone back. First time doing this. Now I'm thinking, oh, we should all see this. Hey guys, you should see this. Now I'm standing back here. You know, I'm going. It dawned on me I'm going over it with all of these guys, and you might not hearing it. Okay, now let me squeeze back to the front. Thank you. All right, so what we did, the machine ran it first through this first bath. This is the crystal violet. Crystal violet is the first step. It stains all the cells purple. Then it ran it over here to a rinse cycle, and all of these are timed. So we program the machine to tell it how long, and if this doesn't work, we can tweak the program. So you guys are our guinea pigs. We're going to see how this comes out. And if any one step is maybe too long or too short, then we can come back and fix it. So after it rinsed the crystal violet off, then it dipped it in the second bath. This is the iodine. What iodine does is iodine and crystal violet chemically react together, and it forms that second step is called the mordant. And so what will happen is iodine complexes with crystal violet, it forms a larger structure, and it locks the stain into any of your gram-positive cells. Any of your gram-negative cells, you've got this large structure, but it's just sitting on the outside of the cell. It's not inside of the cell structure. So now what would happen is now we're rinsing off that iodine. The third step is the most critical step in the gram stain, and that's the decolorizing step. This is where the acetone alcohol is. And what's going to happen is acetone alcohol will completely decolorize any of those gram-negative cells. So those gram-negatives where the crystal violet iodine complex is just sitting on the outside of their cells. The acetone alcohol will just rinse that away. As far as your gram-positive cells where that complex kind of soaked into the cell wall structure, the acetone alcohol actually dehydrates the cell wall, locking it in, so actually ensuring that the crystalline remains. So if we were to stop it at this point, and you were to throw it on your microscope, the Staph aureus would be purple, and your Pseudomonas aeruginosa would be colorless right now. So this is the step I'm worried about. It's a really fine-tuned step that I don't know how a machine can do it, because everyone's smear requires a different amount of decolorizing. So we set it for 30 seconds. So now it's going to rinse that off, because as long as that acetone alcohol is sitting on the slide, it continues to decolorize. Once you rinse it off, it stops the decolorization process. too much can actually decolorize a gram-positive cell, and you can end up with colorless gram-positives. So if we have no color on our slides at all, we will be turning that down. We'll be going down to 20 seconds, see if 20 seconds is sufficient. We'll be grabbing your replicates and we'll throw them through. So now it's just rinsing the acetone alcohol off. Isn't that cool? It's just a thin balance. Alright, now it's going to come in and it's going to do the last, what we call the counter stain, which is the safranin. So because your gram-positive cells should all be saturated with the crystal violet iodine, safranin is not going to stain them because they're already dark purple. But because the gram-negatives are colorless, they will soak up the safranin and they're going to become red at this point. And that's how you differentiate a gram-positive from a gram-negative. And I haven't done cell wall structure with you guys yet, but in class we'll actually look at cell wall structure, and I'll go through this again to explain why gram-positive retains the stain and why the gram-negative doesn't. Pretty cool, huh? So again, the reason we switched, most hospitals are running automatic stainers now. Very rarely do you see manual staining being done. Most clinical practices are running these. So it was just time for you guys to be able to get with the time and become automated.
Especially by the time we're through nursing school.
Jeez, this could be archaic.
This might not
You
even
may be
be used
looking back
anymore.
at this thinking, I remember when we used to use these automatic stainers. Alright, so now it's running through the safranin.
Yeah. Back in the day, we used to use spit.
Yeah. Mode of the future, what we're really going to see eventually is computers, and you're going to take a patient blood sample, you're going to put the drop onto the computer, load a tray in, it's going to genetically analyze that blood sample, separate out what's human, what's bacterial, what's viral, what's fungal, and it'll come back with a genetic readout telling you exactly what's in that blood sample. It's going to be different. But that's not there yet. That's a future project. Alright, so there's the rinse, and then after this it's going to air dry, and we can come get it. So now, you've pretty much seen it all. It's going to go into this tray, and we've got a fan in there that's going to air dry it. It sits for a minute. So why don't we go look at your cultures, and then we'll come back and grab these? Or I'll come grab these, and then you can come get them from me.
I just want to see if we're in the
Yeah, go right ahead, absolutely. I want to start troubleshooting your 3 pattern, 4 pattern, 6
I'll take pictures of those steps.
And that is how you stain the slides. Polystainer 2015.